So it turns out that my original video tutorial was so popular that I decided to make another video to help you work out these chromosome counting type questions or problems. And I'm calling this more counting chromosomes, tricks of the trade. So really what it comes down to in a lot of ways is that you have to be very clear about what the form of the chromosomes is in each phase of cell division and what the ploidy of the cell would be in each phase of cell division. If you're very clear about that, you can solve these problems, these word problems, much more easily. If you're not clear on it, then it's practically impossible to get the right answer. So what I did was I created this slide, but I'm leaving it in the edit mode so that I can fill this in. This is about the cell cycle, and of course including mitosis, and this is the image from your textbook. But I just made up a little grid here, and we can fill in what the chromosome form is in each stage, and then what the ploidy of the cell would be in each stage. So in a G1, the chromosomes are in the monad form. In S phase is when they transform from monad to dyad, because that's when DNA replication happens, and DNA replication is replicating the monad chromosomes, and so they're in the dyad form after that's completed. So by the end of S phase, you have dyad chromosomes. Dyad chromosomes continue through all the phases until you get to anaphase. Anaphase is when the dyads are pulled apart and you go back to monad. And then in telophase, you still have monad. And then a telophase cell, at the end of telophase, then each of the daughter cells can go back into G1. So you can see where the transition points are in S phase and in anaphase. The ploidy of the cell is a little different. Whatever the ploidy of the cell is stays pretty much the same throughout the cell cycle. Most cells are diploid, so if you start with a diploid cell, it would still be considered diploid. The only time when you have a change to that would be in anaphase. I guess you could say that the cell becomes tetraploid, maybe, or we say that the number of chromosomes doubles. But that's mainly an artifact of the fact that the dyads are now, each one dyad is pulling apart and is now counted as two separate monads. So, but I guess technically you could call it a tetraploid cell. And then, I don't know why this is going off. Here we go. And then by telophase, by the end of telophase, I should say, you're back to your original ploidy. So this would be a good example. If the cell was diploid, it stays diploid, and the only time that it changes would be anaphase, when the chromosomes are pulled apart and you get that doubling of chromosome number. You could start with a monoploid cell and it would be monoploid all the way through except at anaphase. It could be considered diploid. I mean, it's just that at anaphase you have that doubling. Okay. Now, when we go to meiosis, things are different. And you know meiosis is different. In meiosis, typically there is going to be a DNA replication that precedes meiosis. So by the time we get to prophase 1, the chromosomes are all in the dyad form. And they stay in the dyad form even through anaphase 1. And they don't get pulled apart until anaphase 2. So at this point, you have the switch. And in telophase 2, they are monads. So you don't see that switch till the very end of meiosis 2. So you have to remember that. Now, ploidy is different. In prophase 1, let's say, assuming you're starting with a diploid cell, which is common, then that cell remains diploid. Even in anaphase, it would have to be considered diploid. And by the end of telophase 1, in other words, when cytokinesis is finished, each cell would be haploid, which in this case would be monoploid. Then the cell remains haploid for the rest of the phases, except in anaphase 2, when the chromosomes are pulled apart. Remember that the chromosome number doubles. So you could say that you go back to a diploid number of chromosomes during anaphase 2, but when the cells separate by the end of telophase 2, each cell is now haploid. So then I wanted to go through some questions that are a little bit harder than the ones in the first tutorial. It says, what is the diploid number of a species if a cell from that species is in metaphase 1 and has 16 tetrads? What you're going to do with this is you need to think about, okay, metaphase 1. What's the form of the chromosomes and what's the ploidy of the cell? Well, the form of the chromosomes in metaphase 1 is still dyad. So dyad chromosomes, and you have a diploid cell. Typically. A diploid cell is always kind of the assumption unless they tell you something different. You can default to a diploid cell. Now, if you have dyad chromosomes in a diploid cell, really what you need to know, since this is a diploid cell, you just need to know how many chromosomes are in this cell, and that would be your diploid number of the species. Well, it has 16 tetrads. 16 tetrads would be how many dyad chromosomes. So the question here really is 16 tetrads equals how many dyad chromosomes. Remember that a tetrad contains 2 chromosomes. So 16 tetrads would be 32 chromosomes. And so there's my answer. But I've already worked that out for you. All right, let's go on to another one. What is the diploid number of a species if a cell from that species is in prophase 2 and has 14 chromatids? Well, prophase 2 is a haploid cell, and it has dyad chromosomes. Now, if we figure out how many chromosomes are in this cell, then we would need to double it to get the diploid number, because this cell is haploid. But how many chromosomes is 14 chromatids equal to how many dyad chromosomes? Remember, a chromatid, there's 2 chromatids in a dyad. So 14 chromatids would be 7 dyad chromosomes. But since this is a haploid cell that contains 7 chromosomes, then the diploid number would be twice that, so it would be 14. So the diploid number would be 14. So you must be clear about what your form of your chromosomes is, because that matters in some of these questions. And you have to be clear about whether the cell is haploid or diploid. If you can do those things, you should be able to solve these problems.